Bismillahirrahmanirrahim. Assalamualaikum. For this project, I focus on the valorization of locally algal biomass for a slow-release biofertilizer production. Modern agriculture still relies heavily on synthetic NPK fertilizers, which are known to degrade soil health over long-term use. At the same time, excessive fertilizer application contributes to nutrient runoff, which leads to harmful algal blooms or HABs in freshwater bodies. HABs are not only an environmental issue but also create a recurring waste management problem because large amount of algae are removed from lakes and eventually discarded. In Malaysia, this discarded algal biomass is largely underutilized despite being rich in nutrients. Therefore, the study proposed a sustainable approach by converting freshwater algal biomass into a biofertilizer which is more eco-friendly, supports sustainable agriculture and aligns with the concept of waste valorization. There are several key problems addressed in this study. First, harmful algal blooms are recurring, especially in eutrophic lakes, creating continuous biomass waste. Second, discarding algae increases the risk of eutrophication and environmental pollution. Third, there is a lack of valorization approaches for freshwater algae, particularly in agriculture. Finally, there is limited understanding of algae-based biofertilizer, especially in the local Malaysian context. This gap highlights the need for research that links environmental management with sustainable crop production. So, based on this problem, 3 research questions were asked. How can algal biofertilizer be produced from dried algal biomass? What is the nutrient composition of the formulated algal biofertilizer? And is the algal biofertilizer more effective than commercial fertilizer in promoting plant growth? There are 3 objectives in my studies. The first one is to formulate algal biofertilizer using dried algal biomass. The second is to determine the nutrient composition of the biofertilizer and to evaluate its effectiveness on latest growth, comparing algal biofertilizer with commercial NPK fertilizer. So, this research is significant in several ways. It supports sustainable agriculture by reducing dependency on chemical fertilizers. It also promotes the reuse of waste algal biomass. And it also aligns with SDG 9 and SDG 14. Right, so we go to the overview of algae in our literature review. Okay, algae are aquatic photosynthetic organisms ranging from microalgae to macroalgae. They differ from the plant as they lack true roots, stems, and leaves but possess diverse pigments that allow them to adapt to various light conditions. Despite their simple structure, algae are nutrient-rich and have strong potential for agricultural application. So, what is harmful algal bloom and what is the causes? Harmful algal blooms occur mainly due to nutrient enrichment, particularly nitrogen and phosphorus from agricultural runoff. These blooms are further exacerbated by heavy rainfall, warm temperatures, and strong sunlight. HABs negatively affect water quality, aquatic life, and increase management costs, making them a serious environmental concern. This is why we need algal biomass valorization. It refers to the conversion of waste algae into value-added products. This approach supports a circular economy and sustainable agriculture. Algal biomass can be reused as soil conditioners, biochemical resources, and biofertilizer, thus reducing environmental waste while adding economic value. Algal biofertilizer are rich in macronutrients such as nitrogen, phosphorus, and potassium, as well as micronutrients. They help improve soil fertility, soil structure, and microbial activity. Importantly, algae also exhibit a natural slow-release nutrient mechanism which reduces nutrient leaching and environmental solution compared to synthetic fertilizers. Therefore, algae is a suitable ingredient for slow-release biofertilizer, or I call SRB. It releases nutrients gradually, matching plant demand. They are biodegradable, improve soil moisture retention, and significantly reduce nutrient runoff. In this study, composted algal fertilizer function as a natural slow-release biofertilizer. Despite extensive studies on algae, there are clear research gaps. Most Malaysia studies focus on biofuel antioxidant or pharmaceutical applications, but not on agriculture. There is limited research on local Malaysian lake algae used as biofertilizer. There is also a lack of region-specific studies on litter growth for Malaysian climate. Therefore, this study aims to address this gap. So, for the methodology, firstly, the algae will be manually collected from a local eutrophic lake somewhere near here, Shah Alam or Puncak Alam. The collected algae will be filtered, dry and ground into a fine powder. The algal powder will then be used to produce two types of fertilizer, which are solid composted algal fertilizer (SCF) and liquid fermented algal fertilizer (LFF). Then, the nutrient content of the fertilizers and soil will be analyzed using an NPK test kit. A pot experiment will be conducted using Electrica sativa, also known as lettuce plant. There are 4 treatments, which each treatment has 3 replicates, giving a total of 12 plots. Plants will be measured on day 7, 14, 21 and 28 and harvested on day 30. For control, there is no fertilizer added to the plant. As control fertilizer, plant were being grown with commercial NPK fertilizer. For SCF, plants will grow with my fertilizer and also the same as LFF, but it need weekly application. During plant growth, these are the aspects that will be measured, which is plant height, number of leaf, SPAD value, which is the chlorophyll content in the leaf, soil nutrient content and pH. Therefore, there are expected outcome on plant growth performance. The LFF is expected to show strong early growth with taller plants during weeks 2 to 4. The SCF may show slower initial growth but faster growth later due to its slow release nature. The commercial fertilizer may produce the tallest plants yearly compared to LFF, while the control is expected to show the lowest growth rate and biomass. However, there are some limitations and risks, and with mitigation plan for them. I can say that pot experiments may not fully represent the field conditions. So, the soil amount, pot size and watering will be standardized for control conditions. There is also a risk of microbial exposure during fermentation. Therefore, it will be managed by wearing gloves and working in well-ventilated area. There is also a risk of phytotoxicity to plants, which may inhibit the plants. Therefore, plants will be monitored regularly for symptoms such as wilting or yellowing. Plant disease and pest attack will be reduced by sterilizing soil and using physical barriers like nets and fan. Next, this is my Gantt chart and the reference list for this slide. In conclusion, this study explores the potential of local algae as a slow-release biofertilizer. It also contributes towards sustainable agriculture, eco-friendly farming practices. Thank you.